It is a pleasure to introduce today's seminar speaker, Dr. Bushra Khan. She is a project scientist in environmental toxicology at UC Davis. She also serves as laboratory director and quality assurance officer at the Marine Pollution Lab at Granite Canyon, which is located on the Big Sur coast. Her work supports a statewide monitoring program for the California State Water Resource Control Board and focuses on advancing environmental monitoring through toxicological tools and collaborative cross-sector approaches. Her current research includes identifying patterns and effects of contaminants in water bodies across California through environmental monitoring and toxicological evaluations. She holds bachelor's and master's degrees in zoology from the University of Delhi and earned her PhD in biological science from UNC Charlotte, where she studied biomarkers in estuarine bivalves. She went on to conduct her postdoctoral research at the US EPA and the Oak Ridge Institute, focusing on emerging contaminants such as nanoplastics, pharmaceuticals, and PFAS. Please join me in welcoming Dr. Bushra Khan, whose talk is titled Integrating New Tools and Approaches into Environmental Monitoring Programs in California. Thank you so much for being here.
Good afternoon. Can everyone hear me at the back? Okay. Thanks for having me. Thanks for hosting me. I am thrilled, beyond thrilled to be here. I am particularly excited because I feel a lot of folks, just generally who go to UC Davis or belong to UC Davis, do not know that our laboratory is part of UC Davis, part of Department of Environmental Toxicology. So I'm excited to share all the work that we do with different state agencies, and I'm hoping that this visit is a beginning of a partnership and we can find collaborative projects and meaningful engagement with Bodega Marine Lab as well as UC Davis Department of Environmental Toxicology. Okay, so I have a lot to share, and for people who did have meetings with me this morning, they know that I can talk a lot, so I will try to make this simple and quick, and I hope that there is a discussion at the end. So I hope to spend at least 10-15 minutes for some questions at the end. So I've broken this presentation into 4 big blocks. So I'm going to start with some introduction and background of the laboratory and the kind of work that we do. And then I'm going to introduce the monitoring program that we run for the State Water Resources Control Board and what we are currently doing with the program. This is an 18-year-old program. And then I'm going to go into the new tools and approaches that we're trying to integrate into that monitoring program and share some of the ongoing work at the very end of this presentation. So let's start with the introduction and background. So our laboratory is about 4 hours south of Bodega Marine Lab, and we are about 3.5 hours away from UC Davis. It's sort of a triangle. You guys are up here, about 2 hours from UC Davis, and we are down here. So because of our distance from the Bodega Marine Lab as well as UC Davis, we don't get a lot of opportunity to engage and partner with folks on campus, which I hope will change. But over the years, the laboratory has conducted a lot of different kind of water quality and monitoring work, and there have been academic partnerships as well with folks on campus. So this is what the lab looks like now, and this is what the lab used to look like in the 70s, I want to say. So this laboratory was a Navy facility in the 40s and the 50s, and in the 60s, NOAA, the federal agency, took over the facility. And there was a lot of aquaculture work that happened after that time, which belonged to the California Fish and Game, which is now known as California Fish and Wildlife. And along with being able to work on the intersection of academia as well as academia and regulatory sectors, one of the perks of being at a place like Granite Canyon is the awesome views, which you guys also have here. So I think I always, whenever there is a new person coming to our lab, I always invite them to come see us, see how isolated we are, because we are 8 miles south of Carmel, so we are in the Big Sur area, but also just to be mindful of where we are located, and we have access to pristine seawater, so there are a lot of different kinds of experiments that can be planned and conducted. So a little bit about the history. So we have a rich history of collaboration with different government agencies, I mentioned, regional and academic partners. And over the years, we have done different kinds of work. There was some oil dispersant work. There was a vector study, a lot of vegetated ditch efforts, agriculture and urban land use work, water quality, toxicity, identification, evaluation. So there's a rich history of the kind of work and the kind of partnerships we have had in the past. And then specifically due to our unique location, we also have partnerships with several different agencies and groups who take data on weather patterns, who are looking at the ocean temperatures, and then NOAA, who owns the facility, also conducts their gray whale census at a facility because of the uniqueness of where the facility is located. The gray whales, during their migration south, they come closest to the shore. So they are there every year to conduct their survey. But if I could summarize everything that we have done in the last 3 decades or more, is that most of our work is geared towards informing state, regional, and local resource management programs, including the Clean Water Act and the 303D listing of impaired water bodies of the Clean Water Act, as well as the 305B condition assessment and total maximum daily load. So all the work that we are doing, the purpose of that work is to inform monitoring programs and managers to make the right decisions to protect ecosystems. Okay, so there's a lot of standard toxicity testing at the lab that has been done over the years, and I don't need to explain what standard toxicity testing is to this group. So we measure toxicity of samples using standard test organisms, and most of our work is on environmental samples. So we go out to different places in California, all across the state, and collect environmental samples. And sometimes some of our projects are not directly on the environmentally collected samples. They are on samples that we prepare in the lab to simulate an environmental condition. So currently we have a project with California Division of Fish and Wildlife where we are creating the high energy water accommodated fraction with renewable diesel to simulate how, if there is a spill in the environment, how the animals would be exposed to it. But majority of the work is on environmentally collected samples. With those samples, we conduct different water and sediment tests using acute and chronic protocols, and these protocols are all standard EPA tests. And we measure standard toxicity markers like survival, reproduction, development, and growth. And on the bottom right, you have the Hylella, which is our poster child of the test organism that we use. So that's an amphipod, a freshwater amphipod. And most of the data that I will be sharing with you today would be on the Hylella test. Currently, so there are a lot of test organisms that we use, but these are the current test organisms that are being used for different projects. We are, as I mentioned, we work with Hylella. There is Chironomus dilutus, which is our midge larva, which is the second most studied organism in our lab. We do a lot of water tests with Cerodaphnia tubia. We have some fish tests right now for our CDFW project, both freshwater and marine fish. And then we do some larval assessments using Mytilus as well as sea urchin. So using multiple species is common sense in the world of toxicology, but it should be reiterated here again because standard toxicity testing is used by regulatory agencies, but it always does not have the sensitivity that we need. So we need to use multiple organisms and multiple markers to expand the sensitivity of the tests that we are doing on these environmental samples. So this is one example of a data set that is very relevant to the monitoring that we're conducting for the state. So in the first column, you have these 3 pesticides And these pesticides we find in the environment at multiple sites. And the tests that we have done with the samples that do contain these pesticides, they're conducted with different organisms so that we are able to capture the toxicity fingerprint of the sample using organisms which are sensitive to different magnitude to different contaminants. So for example, for something like bifenthrin, we see Hylella is very, very sensitive to it. So these are LC50 values using 96-hour water test. And for something like fipronil, we see that Chironomus is very, very sensitive. So those are the kind of considerations that we think through when we are designing a monitoring plan for a statewide program, because different sites are going to give you a different chemical soup. And if you're going to be using standard toxicity tests, you want to make sure you're not using just one animal, one marker, because then you are shrinking the toxicity assessment of your sample. Along with using multiple species, of course, we have to use multiple markers. Now, in standard toxicity tests, mostly what you do is kill them and count them, right? So you're looking at survival. And those are the protocols which EPA uses, which are of regulatory relevance. And along with that, you can use some sublethal markers such as growth. And growth has been very, very useful for the monitoring program that we administer. So this is data from last year, 2024. This is amphipod survival. And using survival, we were able to identify, 8 sites, I think, using just the survival marker for the amphipod. When you add growth, you really shoot up your identification of toxic sites. And that is very important for us because without that marker, we completely lose more than half of the toxicity information. When you add another organism, so this is the MIDGE, the Chironomus dilutis toxicity test, addition of Chironomus identified 3 unique sites. Now, 3 does not sound like a lot, but when you are working with 90 sites and one marker with one organism identifies 9 sites, the second marker with the same organisms adds 28 unique sites, finding additional 3 sites which are toxic, which can inform the state about what water bodies are impaired is very important. So we do with standard toxicity what we can with the given resources, but definitely there's a need to expand the sensitivity and relevance of the tests that we are conducting. And doing that is hard with standard toxicity tests. So one thing you can do is you can add more animals and more markers using standard EPA protocols, but these tests are very expensive to run. And so when you're running within the budgetary constraints of a monitoring program, you have to become innovative. And you have to bring some of the sublethal toxicity marker information, which is not standardized yet, into the existing framework of the program, which is what I'm going to show in my new tools and approaches section today. So there are many other tests. So these are some examples that I used for introducing what we have been doing at the facility, but there's a whole catalog of standard toxicity tests for ecological risk assessment that EPA has. And they definitely, as I said, give you a wealth of information, but are resource intensive. And to be able to run these tests for a regulatory agency, the lab needs to be accredited. So which is a whole process to make sure that you are running the test right, there are audits included in that. So maintaining all of that is very resource intensive. And all of these tests, they can help solve complex or inform you about complex environmental challenges. So a lot of what we do is monitoring, and that's just a statewide annual monitoring program, but we have also done work with, for example, post-fire samples. So the same tests, because they're so standardized, can be used to understand the effects of disaster, the effects of disaster mitigation. You can look at oil spills and look at the same test and contextualize it based on the chemical analyses that you can get from these different kinds of disasters. And you can also use these toxicity markers for different kinds of pollutants. The important thing to remember here is that although standard toxicity testing is fairly simple to conduct, tedious but simple to conduct, you need to pair it with contaminant analysis to evaluate these very complex problems. And what you have to do with those data sets is contextualize it with the problem at hand. So for our monitoring program, the most important thing is land use. So you take the standard toxicity test that we conduct, and then the contaminant analysis that is done at an analytical laboratory, and contextualize those data within the realm of land use in California. Okay, so that brings me to our monitoring program. So the monitoring program is called Stream Pollution Trends Monitoring Program. A little background on that: it is a program of the California Water Resources Control Board. And under the umbrella of water boards, there is another larger monitoring program, which is called the Surface Water Ambient Monitoring Program, which creates data for managers, decision makers, and the public, and gives high quality information to evaluate the condition of all water bodies throughout California. Under that program, there are 4 statewide programs, and stream pollution monitoring program that we administer is just one leg of those 4. So for the program, we have been going to up to 100, but right now we have 90 sites all across California since 2008. So the program is in its 18th year. And for all these years, we go to the set sites, we decide what sites are important, and that is based on how the watershed is draining. And you decide on what would be a representative site to understand the toxicity of deposition of sediment. The toxicity testing is conducted at our laboratory, and the same sample that you collect from the sites is also sent out to a commercial lab for contaminant assessment. And the data feed into California Environmental Data Exchange Network, or CEDN. And there is also a public-facing data dashboard where you can look for a particular site or particular contaminant or particular toxicity test, and all the data would be plotted for you, and you can download it. And the sites that we have, they cover all 9 regions of California. So under the big umbrella of the California State Water Resources Control Board, there are 9 regional boards. So our data serve all those 9 boards. And as you can imagine, California is diverse. So when the field crew is out collecting samples, sometimes there are beautiful places, and sometimes there's just trash. And a lot of this work is pretty challenging, and it requires quick decision making, because you're crawling under bridges, you're collecting samples in areas where you have to assess safety. You have to make sure you're collecting depositional sediment, because you want to only collect depositional sediment because that's representative of the transport of sediment in the previous year. So there's nuance to it, but we are able to collect data that represents so many watersheds and sub-watersheds in California. And a lot of these data, even though they are produced for the monitoring program of the California State Water Resources Control Board, they are used by several, these are just some examples, several different state agencies, including DTSC, DPR, the Division of Water Quality's Contaminants of Emerging Concern Unit, so, and the public. So anyone can access these data. So we want to make sure that data are high quality. That's why we need to be an accredited lab, but we also want to make sure that we are providing the state with maximum information about the toxicity or potential toxicity of these samples using the money that we are given. So all the work that we do for the program, that is geared to answer 4 big management questions, which are very long, multiple bullet points. So I've broken them down into 4 cute graphics. So the first thing the first management question has to do with trends. So the state wants to know the trends of contaminants and the trends of toxicity in the context of land use in California. The second one is impact on beneficial uses. So again, the state wants to know if at these 90 sites, the beneficial uses of these water bodies are being affected. And beneficial uses is a regulatory term. There's a whole list of what is considered beneficial uses, increasing recreational activities, agricultural use, swimming, all kinds of things. The third question is about causes. So if we know the sites are toxic or the sediment collected from sites are toxic, what are the causes of that toxicity? And the last one is regulatory action. So if the state does take a regulatory action, they want to know through our monitoring program if those regulatory actions are effective. So there's a lot to unpack there. So for today's presentation, I am only going to focus on two boxes, so the trends and the causes. And the story that I'm going to tell you about a particular contaminant and about new approaches is going to focus on trends and causes. So because the questions that the management questions that the program is aiming to answer, they are multi-layered and they change. The design of this program, of course, needs to be very adaptive. And that the fact that the design needs to be adaptive is where you can leverage some of the tools and techniques which are still not standardized, but you have to marry those new tools and techniques with the existing framework of this program. Okay, so I have been told that whenever I introduce the program, I forget the most obvious thing, which is that SPOT is a trends program. And I don't usually remember to mention that because the acronym has it. It's called Stream Pollution Trends Monitoring Program. But I think it's important to mention that SPOT is a trends program. So we are looking for patterns. We are scanning for big trends and big patterns and patterns over time. So we look at temporal trends, but these trends can be analyzed at several different scales. So one of the scales that you can look at these trends is a statewide trend. The second one is the land use, which I've said many times because land use is very important to the state. And the third one is across sites. And they're color-coded because they will make an appearance in many different slides on this as PowerPoint. So those are the scales that we are working at. And the trends are identified, just to remind everyone, for the sediment contaminants as well as sediment toxicity markers. So this is just, again, a reminder, the samples that we collect go through collection processes in the field, then they are loaded in different tests, and they're run through many different tests. For today's presentation, we are only going to talk about the HiLALA test. Okay. Very busy slide. This is just a snapshot to show what we have done in 18 years. So on the very top, just to orient everyone, on the very top row, you have all the years. And the first column has all different kinds of analyses that we have done for the program. And you can see at the very top where you have number of sites, the sites go up and down in number a little bit, and that has to do with budgets. And then below that, you have two rows of toxicity tests. So the first one is HiLALA, which is what we're going to talk about today. The second one is the MIDGE. And you can see the MIDGE was not added to the program up until 2013. 13 and 14 data are missing, but it was added in 2013. And the reason to add MIDGE was there was a particular kind of pesticide that HiLALA is not very sensitive to. So to be able to identify effect of that pesticide, we had to add another organism. And then everything below that, all these rows show different kind of contaminants that we have analysis on for all these sediments collected over the period of 18 years of the program. So for all these data, to work through all these data, it's very important to remember that California is a very large state. And there is a lot of spatial diversity that we are working through if we really want to understand or contextualize these data in terms of land use. So up here on this map, you see different land uses. The green is agriculture, the red is urban, and then there are open and other kinds of land uses. And the two important land uses that we are interested in are urban and agriculture. The second graph, or the second map down here, shows you all the sites that we go to. And if the dot is red, that means it is urban influence site. If the dot is green, that's a agricultural influence site. And it's important to remember that the diversity of land use is also seen in the diversity of toxicity profile. So again, very busy heat map, but all I want you to focus on this heat map is there's yellow, there's red, and there's green. So green is not toxic to HiLALA. Yellow is moderately toxic and red is very toxic. And we are only talking about one organism, one marker. Every row is a site and every column is a year. So if you just, I have to move so I can see. If you look through something like this, if you move your eyes from left to right, you see the site was moderately toxic and then became very toxic. For some other sites, they are never toxic for this organism and for this market. Doesn't mean it's not interesting. It's just not interesting based on this data set. So this diversity of land use and diversity of toxicity profile is very important to keep in mind. And also remembering that when you look at everything on a statewide scale, a lot of little pieces of information get diluted. Because if you're looking at medians for toxicity or medians for any contaminant on a statewide scale, because California is so large, you're going to lose trends that are happening at small scales. So even the state is interested in statewide trends, which is important, we still produce that. It's important to remember, just because you don't see a trend on a statewide scale, it doesn't mean there is no trend at all. Scales matter when you're looking at data on such a large magnitude. And that's why regional or site-specific scales are very informative. And we have been saying this to the State Water Board that we should start looking at things, not that we are not already looking, but for this program, maybe pooling the regional trends are more important, because some regions are way more urban than others. And this kind of diversity, we also see in contaminant profiles. And the dilution, so to speak, the dilution of statewide trends is seen when you look at contaminant profiles. So on the first column here, you see different kind of contaminants, except for the metals at the bottom. Everything else is a sum. So each of these contaminant class is the sum of many different analytes. So already you're pooling things. So you're looking at contaminants at a different scale. The second column here is a statewide trend. And you see that most of these things are stable or nothing is happening. And if you were to just look at that, you'll say, okay, then nothing is happening for pyrethroids. That is not correct, because there's a lot that's happening pyrethroid. You have to just adjust your scale or fine tune your scale. And the last column here is the most interesting one. And this is why the state is so interested in land use relationships, because as you can see, most of the contaminants that the program has measured over the years, they are significantly correlated with urban land use. So this is just to show that the trends and correlations are scale specific. And the same thing you can do with toxicity markers, right? So when you look at toxicity markers, we don't see for HiLiLa any statewide significant trends, but we see for both mortality and for growth, both of those markers for the amphipod, significant correlations with urban land use. And that is important because, just to put it in visualize it, over a period of 17 to 18 years, when we look at the percentage of sites which show increasing trends of toxicity, you see the tan and the orange wedges here, which amount to be 10%. You see mortality of this particular amphipod increasing. And for growth, it's even a larger proportion. So again, 10% doesn't sound like a lot, but remember, you're looking at data over a 17 or 18-year period and looking at that and seeing if the mortality of this organism is going up, which means some of these sites have been consistently toxic, and we have to find what is causing that toxicity so the state can implement some regulatory action and then continue monitoring for this so that we can tell the state if the regulatory action really had an effect. Okay, so the big question, I think by now I've mentioned land use like 8 million times. So I think it's very clear that the land use portion of this puzzle is very important. And what we want to know, one of the key questions of the program is what are the drivers of toxicity in urban areas and use in urban land use? And one of the drivers is the pyrethroid pesticide. So I'm going to tell you a quick story about one of the most notable findings of our program. And I call it the urban story because everything comes down to the use of this pyrethroid to urban use. And when you look at pesticides, your first instinct is, oh, it's coming from ag. And through 18 years of data that we have to show for the program, the pyrethroid pesticides are causing toxicity in California sediment, freshwater sediment, but they're not that they're not coming at all from ag, but the majority of the contributions of pyrethroid pesticides are coming from commercial applications in urban areas. Okay, so I'm pretty sure everyone here knows about pyrethroid pesticides, but pyrethroid pesticides are insecticides. They are used very commonly. You can go to Home Depot and buy it. It's in Raid. It's in different kind of pest applications and killers. And I'm going to give you like a very zoomed out summary or version of this. On a statewide scale, we found through the years that the toxic potential of pyrethroids remains high. So on this graph, what you see on x-axis is years 2010 through 2022. And on the y-axis, you see a mathematical calculation for a toxic unit. What that basically shows is the toxic potential of these contaminants. And the red line is where your concern should start rising. Everything above 0.5 toxic units is where you should have some concern. Everything above 1, theoretically, is going to kill 50% of the population. So statewide, on a statewide scale, even though we see a lot, for what I explained in some slides ago, is at a statewide scale, a lot of times you miss trends. But the pyrethroid problem is so pervasive in California that on a statewide scale, we can still see the toxic potential of this contaminant increasing. And when you map the toxic potential, just to visualize it spatially, you see these big red dots where you have a lot more pyrethroid present, mostly are in urban areas. This is, so you see San Diego, LA, and in the Bay Area. And you also see for most of the sites we go to, which is 90 sites, you have a trend of increasing pyrethroids over the years. And I think, again, it's important to mention that the trends are measured over such long time periods. For them to continuously show that they are going up just shows you the magnitude of the problem with this pesticide. Okay, so we now know that the pyrethroid toxicity potential is increasing in California. And when you look at it from the land use lens, you see that wherever you have increasing urban land use, when you have increasing urban land use, you have increasing pyrethroids in the sediments deposited year after year at these sites. And these 4 bars, they show different bins of land use. So the first is 0 to 25. This is 25 to 50% land use, 50 to 75, and 75 to 100. So with increase in urban land use, you have increase in pyrethroids. But also with increase in urban land use, you have decreased survival of this test organism. So all in all, through different modeling and calculations, the story here is that with increasing urban land use, you have increased pyrethroids in the sediments, which are coming from urban applications, which increases the potential of toxicity from pyrethroids, which leads to decreased survival or increased toxicity for this organism. And even though this is a great story and an important story to tell, there is still gap between toxicity and targeted chemistry. So we found so many sites where pyrethroids are causing toxicity. But the question after you have answered or you have told the pyrethroid story is, do pyrethroid pesticides explain all of the observed toxicity in California? And the answer to that is no. So we know pyrethroids are part of the problem, but we have a set list of targeted contaminants, and we measure the same contaminants over and over again every year. So are we capturing all the contaminants? And of course, the answer to that is no. Are we capturing all the toxicity? The answer to that is also no. So how do we modify the list of priority contaminants? For that, we need to understand what's in the water or sediment. And finally, the current toxicity markers that we are using, are they adequate to capture the toxicity of these contaminants or the threat or the dangers associated with these contaminants? Because remember, I only showed you data for one organism. We have another organism, but a 2 organisms with 2 markers enough to capture the toxicity profile? No, they're not. So we have to use some other tools to expand the sensitivity and the relevance of our program and or the data that we are uh, providing to the state. Which brings me to my most interesting section, the new tools and approaches that we are integrating in the program. And before I go into the new tools, I do want to mention that the tools and approaches, these are not there to replace the existing tools. These are there to augment and to expand the scope of the existing tool. Also, none of these tools are standardized. So we have to continue working with these tools to show the state and other regulatory agencies they actually work and they actually add value to the program, because these are also expensive to do. I am going to run very quickly through them, but I want to show the kind of tools, how they're used, and the examples. So if anyone has any questions about like the details of the data that I'll be showing, happy to talk afterwards, but I'm going to go rather quickly through these sites. Okay, so the easiest way I can think of or visualize new approaches being integrated into a sediment monitoring program is via the sediment quality triad. So very, very simply, the sediment quality triad takes different kinds of approaches and different pieces of information to assess the quality or health of the sediment. And the 3 big fields that are used for that are chemistry, toxicology, as well as ecology. So the current approaches are using a set targeted analytical approach to look at one or 2 or 3 kinds of contaminants. So these are targeted analytical methods. That is the chemistry piece. The toxicity piece is doing standard toxicity testing, like our lab does, which is what I showed you for the PyTest story, for the other Chironomus, Hylella, those kind of data. And the last one is community metrics, so which I'm not going to go into at all today, but that basically tells you what kind of organisms are in the sediment and what is the proportion of their existence there. So the community metrics is called bioassessment. And right now, the way this is done is you count the organisms and you measure how many of what kind are in the sediment. All of that can be modernized and revolutionized, not by replacing, again, but by augmenting. So for chemistry piece, we can use non-targeted analysis. For standard toxicity testing, we can use this in addition to much sensitive biomarkers or bioassays. And for community metrics, which I'm not going to go into at all today, you can use environmental DNA and RNA. So for the non-targeted piece, I'm not an analytical chemist, but I have taught myself enough to talk about it semi-intelligently. So the non-targeted chemical analyses, they function as a broad screen of thousands and thousands of chemicals. So they have a lot of coverage of the chemical space of your sample. They may not be sensitive enough for each of those chemicals, but you get a lot of information, and then you have to run through that information to extract the relevant pieces. And there are different levels of confidence attached to the data that's coming out, because you may get something that you don't have enough information about. There is no standard to confirm it is X chemical. So you may get a molecular mass, or you may just get a formula. So there are different levels of confidence that you can attribute to this, this kind of analysis. And the bioanalytical or bioassays, biomarkers, those are, these approaches use evolutionary conserved targets. And the power of using evolutionary conserved targets is that you are then not limited to that one organism that was providing you toxicity information. So when you take evolutionary conserved molecular targets and use them in the context of this large chemical space that the non-targeted analyses can capture, then your toxicity assessment becomes exponentially more powerful. Or so we are trying to convince the state. Okay. So in 2021, we did a pilot study. We chose 30 sediments from the 90 that we have, and they were selected for non-targeted analysis. Samples were extracted and analyzed using GC and LC high-res mass spec, and they were library matched to identify different features. I call them features and not chemicals because not all of them are confirmed using standards. So that's an analytical term. The feature is basically a chemical, but you have different levels of confidence associated as to what kind of chemical it is. And after that, we ran Spearman rank correlations, which were drawn using peak heights and the Hylala toxicity marker. And using those correlations, we could decrease that large chemical space to some chemicals of interest. And I'll show you some examples of that. And then features which were related or significantly related to toxicity, they were investigated further using targeted mass spec and quantified using standards. Okay, so our workflow was something like this. We collect environmental sample, run it through the non-targeted analysis and GC and LC mass spec. We are already doing toxicity testing, so that is at no cost. That's already happening. And then we use a prioritization workflow to prioritize the chemicals that have been identified by non-targeted analysis only after they show significant correlations using a rank correlation system. And we also can use these data to do other kind of land use correlations, because you can just take all this data set, run it through a land use correlation model, and find what chemicals are present or significantly related to urban land use. So you can be as iterative as you need to be. You can ask different questions from the data set because the chemical space that you're capturing is so large. And then all of that informs the monitoring design for the future year. So I'm back to my questions of can we use this model to understand or identify the drivers of toxicity in urban areas? And can we use this model to prioritize our contaminant list? Now some really busy slides giving you examples of what NTA has done for us so far. So in our first run, we had the chemical space had about 700. So we've identified 700 features. And after running through different thresholds and our rank correlations, we were at about 45 features correlated with toxicity. And once you start digging into that, you find that some of the features are correlated with Hylala mortality. And it was very validating to see that the pyrethroids were at the top of the list, because we know through our targeted work for 18 years, pyrethroids are driving this toxicity. But we also found some other information that was otherwise missing from the program, such as glycosylide or synthetic musk, some chrysols. Those were also significantly correlated with the toxicity. And then we also used this for our growth marker, found some other pesticides which were significantly correlated. And we do some analyses on these as well, but we have not found any significant correlations until we started using non-targeted analysis. I will mention, though, that just because they are correlated doesn't mean they are the causing factors, right? And then there are other chemicals which were not correlated at all, but we could identify different PAHs, UV blockers, a whole bunch of different chemicals. We also took this data and used the growth endpoint and merged the growth and the survival endpoint into a biomass. So this is growth and this is biomass, and biomass is just a mathematical calculation that you can do using your growth and survival data. And just by changing your toxicity marker, the list of interesting chemicals or significantly related chemicals changed and expanded. So that just shows you the power of using or merging different markers and layering all these pieces of information on top of each other. Okay, same thing with, so that was the GC data, which chemist told me that it's not gas chromatography, it's Gauss chromatography because it's so beautiful. LC, not so much. So LC data are messy, but same concept. We found a lot more, ton more features, total features, and we kept on using our prioritization model to narrow that space down to the very interesting and significantly correlated features, and found some antibacterials, barbiturates, steroids, things that you would expect to be out in the urban spaces. But the program does not have either enough money or interest to look at pharmaceuticals in sediments, because generally they will be present in water. But by using this approach, you can look at some of the contaminants that otherwise would be missed from routine monitoring, such as what our program does. What we also saw is that with increase in person land use, we saw increase in number of features or chemicals, which is not unexpected. When you have more urban land use, you have just more chemicals entering the waterways. So you just see more chemicals in your non-targeted analyses. And what is more interesting than this was expected, but it was validating to see that. What was more interesting than this is that different urban areas were different. This was a finding that we were not expecting to be so different. So there's some principal component analyses that I'm going to show you. Every dot is a site on this PCA, and different colors represent different regions. So most of the dots, they represent the chemical space or the chemical makeup of that sample from that site. Most of them are here, but you see, and these are mortality-related features. You see some which are separated from others. The two on top are region 4 LA. And the one at bottom, which is separated from the main cluster, is the San Francisco Bay. Both very urban, both with similar problems, but clearly their chemical makeup is very different. So the same monitoring design that you're applying to LA, if you apply to San Fran, you're gonna miss a lot of the contaminants that you should be looking at. This is just one example. This is mortality. You add growth to that. Again, we are expanding the sensitivity of our operations. So you see the same thing. Most of the points cluster here, there are some points which are separated, and you see both of these regions separated out, which you see here. Finally, you also see another region, which is also very urban, San Diego. But again, even though San Diego separates out from the main cluster of all other sites, that is very different than San Francisco Bay and then LA. So the point of showing these PCAs is that different urban areas have different complexity in their chemical fingerprint, and they need very focused and different approaches in terms of monitoring the right kind of chemicals. And we, in 2023, so this was 2021, in 2023, we expanded our work and used these correlations for land use. As I mentioned, that is always an option because we have land use data. And we saw for urban land use, 290 features, a lot of pyrethroids, lots of plasticizers, PAHs, and for agriculture, we saw a lot of pesticides. For the toxicity marker, we saw negative relationships, so toxicity survival went down because there were so many chemicals that the animals were being exposed to, and we found 34 significantly correlated features, including plasticizers, phthalates, and bifenthrin, which is a pyrethroid. We also expanded to bioanalytical work where we were doing receptor activity assays, which I'm going to touch base in a second, but I also want to mention that this is a lot of what I'm showing is qualitative. And the state wants hard numbers. They want quantitative data. So what you could do is once you see, oh, there is a phthalate there, or there's a plasticizer, you purchase that. If it library matches, you purchase the standards for that, and you can do a targeted analysis on your sample, which is what we did. So we purchased all these standards and bin them in different categories. And once we ran all these, every pie chart is a site. So once we ran all these samples, we started seeing contaminants that were never measured in these sediments before because they were not part of the set targeted list. But again, it was validating to see the usual culprits. This is pyrethroid bifenthrin, pyrethroid, fipronil, another pesticide. They also showed up. So this is how we show a state regulatory agency that this approach is adding value because it's adding more contaminants of interest. They may not be real causes or drivers, but they should be monitored. Finally, the bioanalytical assessment, we added another layer to this. So we go collect the samples, we do the non-targeted analysis, we do our toxicity testing, and do a biomarker assay. And that biomarker assay is basically a receptor activity assay, which is a relative measure of a sample's potential to trigger a receptor. All of that work together allows us to increase coverage of the chemical space because of non-targeted analyses. And with biomarkers, we find more sensitive markers of toxicity. So the layer right now we have is non-targeted analyses. Then we have our toxicity test, tender toxicity test that the lab does. We add biomarkers to it. And all of those put together allow us to expand the sensitivity of our toxicity assessments. So a lot of correlations have been run through there. And because of biomarkers being so sensitive and this receptor activity being so sensitive to particular classes of chemicals, we have been able to identify different contaminants which are of interest to this monitoring program. And don't have time to go into how the biomarkers are the receptor activities are measured, but these are relative measures. So the way the data comes to us, this is in collaboration with a Dutch lab. We do bioanalytical equivalence concentration using an agonist or antagonist for that receptor. And again, when we lay this, it makes our assessments of toxicity very, very robust. So one of the key findings of the bioanalytical assessment was the AHR, aryl hydrocarbon receptor activity. And when we run some correlations, we found that it was significantly related to bifenthrin, even though that is not the mode of action for bifenthrin. It was significantly related to fipronil, which it may have some effect, as well as PFAS. And I plotted this to point out that there are, in the chemical soup in urban areas, there are so many chemicals and covariables that just because something is significantly associated with a receptor activity assay doesn't mean it's triggering it, but it also doesn't mean that it should not be monitored. So these significant correlations, what they do is you start with a big chemical space and you apply these different correlations to make that chemical space smaller, but better informed compared to what it was before we started using non-targeted analyses. The AHR, or aryl hydrocarbon receptor assay, was also significantly correlated with our organochlorines. Okay. Real quick on our ongoing work, we are continuing doing bioanalytical assessment. We are bringing our estrogen receptor into this picture. So we currently have a lot of different features which correlated with the aryl hydrocarbon receptor activity assay. And if you take your estrogen receptor assay out of the 553 that we initially identified, you get to 312, and then you keep on going down to different correlations. What that does, it answers a particular question. So if the state is interested in the urban land use, you look at this set of chemicals. If the state is interested in a more sensitive marker, you look at these two. If the state only wants to know the high level mortality information, you look at these 25 chemicals and then go by standards for that, run them, and then see if those chemicals explain the toxicity that you are measuring. We do the same thing for LC features as well, and the LC features are much bigger set of chemicals and much bigger much bigger list of chemicals that we have to go through. You can run the same prioritization model for LC as well. So coming back to the management questions, because everything has to fit within the realm of the management questions, what we have done using these new tools and approaches is identified new trends and causes, or potential causes, I should say. We have identified land use relationship with NTA features and investigate different trends. And then for causes, we are analyzing these correlations to make a list of potential drivers of toxicity in different areas in California. And this will help us better inform the state about their priority analytes. We can focus on sites with toxicity correlations. We can come up with monitoring designs, which are very site-specific. But it also has challenges because these are not standardized methods. So it's not like you just go to an EPA protocol and do it. You have to lay the groundwork. You have to write and get approval for the QA/QC measures. You standardize measure, have enough controls, have all kinds of components that go into a standardized test, and then also understand what would be the cost of running the test in addition to standard tox tests. And all of these things that we are doing, we want to make sure that we can corroborate with existing things such as our targeted data sets. And that's like a big question right now that we are trying to answer. Can we get quantitative estimates which are reliable using non-targeted analyses? So we are using this work currently based on our current markers. We are adding more species, hopefully, and more markers. That's up in the air depending on the funding. But definitely adding more sublethal markers in collaboration with Division of Water Quality, Contaminants of Emerging Concern Unit. So this covers a large set of receptor activity assays, and it's really going to expand our sensitivity of the toxicity tests that we do. Then we also want to layer behavioral toxicity on top of this. So we have an EtherVision system which does video recording, and that is a good and environmentally relevant sublethal toxicity marker that we want to integrate into monitoring. And again, more data to identify spatial temporal trends. That means we need to do this work at more sites. But right now, the challenge is we want to continue doing the old framework because that is what the state is required. The state requires us to do. But we want to integrate these new tools and techniques into the existing framework, which of course requires collaborative work and a lot of money. And finally, I'll leave everyone with this. These new tools are new because they are new to monitoring and new to regulatory space. They have been around for a long time. And there was this very interesting piece from a colleague at the EPA who wrote about there's a shift in environmental monitoring, and the time for non-targeted analysis is now. There's a lot of resistance, a lot of questions. But as academics, we have to make sure that this is a tool that can benefit monitoring. To be able to do that, we need to do what we are doing with our program, use it in existing frameworks. And a lot of biomarker work has been going on in Europe for decades, and they are decades ahead of us in using effects-based monitoring and sublethal markers of toxicity. So it's new, but it's new to monitoring. But it's important to remember that there's already a lot of data out there on these approaches, and we should learn and educate our monitoring, or inform our monitoring programs using that. And with that, a lot of people to thank, everyone at Granite Canyon, the director before me, Brent Phillips, the PI, Ron Dema, both of whom have retired, my current team, and then a lot of partners and at the State Water Resources Control Board, specifically Erica Calve and Manuela, who are the leads of the Contaminants of Emerging Concern unit of the Division of Water Quality. All the bioanalytical work is funded by them. All the analytical or non-targeted analysis work is done in collaboration with Tom Young's laboratory on campus at UC Davis. Anna Furyk made most of the figures for non-targeted analysis. She's the primary analyst on that, and also biodetection services or systems who have provided us with the bioanalytical receptor services. Big thanks to water boards and everyone at SWAMP. And if you want to learn more, go check out our website. There's a QR code, and happy to answer any questions. Human. Yeah, I'll be around. Yes.
Looking at the abundance and trends of the biochemistry throughout the Central Valley, it seems like there was a really high, no, it was a high input. There was a really high urban concentration, but the trend going down. Is it just kind of evening out and consistent with the soils, and that's why there's a downward trend because there's less bugs to kill? Or is it more of a knowledge spread with amongst the public, not using it?
So repeat the question and correct me if I got it wrong. So you're asking if looking at the visualization on the map of the pyrethroids in the Central Valley, the trends were going down. And you're asking why is that? Is it because less of it is being used, or is it evening out? Is that correct? Okay. So the pyrethroid use in the Central Valley in non-urban areas are probably coming from agriculture areas. The trends that are going down, they're going like on a temporal scale. So just because a trend is going down, it doesn't tell you anything about the concentration. It's just telling you whatever that concentration is, it's coming down over a period of time. Now, on the other map, you had not concentration, it was toxic units. So which is a mathematical calculation based on the concentration. So in the Central Valley, there are areas where the concentrations are high, but looking from 2008 to now, the concentrations are steadily going down. That's why the trend is down. But the highest, if I were to pick top 3 concentrations of pyrethroids in the state, they are not from Central Valley. So there is still, I'm not saying pyrethroids are not being used in agricultural land use, but they are just they are being used more in urban applications. So, did that answer your question? Yeah. Okay. Yeah. Yes.
What might be driving some of this is what's the average basin of the pollutants from San Francisco versus
So the question is, what is driving the differences between the pollutants in San Francisco and LA or other urban areas? Yeah. It's that's a hard question to answer. So every urban area is different, and a lot of it is dependent on usage, right? So commercial applications, population density, different kind of manufacturing units, that's one part of it. The other part is just the hydrology of the area. How the water is moving through the watershed is completely different in these three areas: San Diego, San Francisco, San Diego, and LA. And so there may be, that's hypothetically, even if there are equal concentrations of pyrethroids, the way they're going to move through the watershed and get deposited at the site, which is our reference site for that basin, drainage basin, is different. So that's one variable. But then again, also, a lot of times commercial application is registered, but household application of pyrethroids is not. So it could be something as simple as that this neighborhood used more pyrethroids than San Francisco used less pyrethroids than LA, something like that. And that goes not just for pyrethroid, that goes for anything. There's more recreational things happening maybe in that water body. There are a lot of moving parts to that, but the simplest answer is cliched, but it's true. Every urban chemical soup is different, and it should be treated as such. So that's why I said when you apply a monitoring design, it should focus on what's in that area. So yeah, that's a puzzle that we're trying to solve. Yes, of course.
I was curious. I feel like there's probably a lot of differences in costs in different areas that have environmental justice equity. So I'm wondering if you are coming from the conservation or whether like an alliance single preference site.
So the question is that different areas may have different uses and different population and demographics, and how is that connected to us relying on a single area to represent that entire area? Is that correct?
Yeah, very this.
Potentially, yes. So answer that question very directly, there are areas where the chemical fingerprint or toxicity fingerprint would be much higher. But the goal of the monitoring program is to come up with a set of sites that adequately represent drainage in different watersheds and sub-watersheds in California. It is very possible that you are separated if it's a point source. Let's say it's a point source, and if you're separated from the point source, by the time it drains and then deposits in the sediment from the site that we are collecting, it may be less. So the question about environmental justice is a different layer, and the state is very invested in that. I mean, they do a lot of work on environmental justice, and that's where the crosstalk between a program who looks at environmental justice and works in that area with a program like ours is very important. But the design of the program and the goal, that's why I kept on bringing the management questions, is not to do that. It's just to represent what's happening in the state and what chemicals should the state monitor and regulate.
Thank
Okay.
you so much.
Thank you for having me here.